Good morning, everybody. Welcome to the WastewaterSCAN partner call. We're going to get started, and my name is Allie, and I'm one of the program directors for WastewaterSCAN, and the other is Marlene Wolf, who's on the call, and many of you have probably heard from Amanda Bidwell, who's also on the call and is a data analyst and helps out on all things related to the project, and we'll all be presenting to you today. Next slide, Marlene. So we always start with this slide, reminding you all of who we are. So WastewaterSCAN is a national effort based at Stanford University to build a national network of wastewater-based surveillance using consistent methods. The program directors include myself and Marlene, who I already introduced. Verily is our laboratory partner, as is National League of Cities for building capacity. And then, of course, all of you are partners from the wastewater treatment plants, as well as state and local and federal public health officials. Next slide. So I'm going to give a program overview, and then we will talk about the H5 marker detection, and we'll end up with an overview of results across the country for SARS-CoV-2 and other targets. At the moment, we have 191 plants participating in the project, and that represents 13.5% of the population. There are 40 states participating in the program, plus Washington, D.C. And this map shows the location of all the plants and the population served. Next slide. We're currently monitoring for approximately 12 different infectious disease targets, depending on how you count them. And if you can count, which I don't know that I can, but we are monitoring for SARS-CoV-2, influenza A, B, as well as the H5 marker, which we'll be talking more about in a bit. That's an avian influenza, RSV, metapneumovirus, and enterovirus D68. We're also monitoring for rotavirus and human norovirus G2. We were previously also analyzing for adenovirus, but we removed that so that we could add another target. And we are also monitoring for Candida auris, hepatitis A, and Mpox. We're doing sequencing of SARS-CoV-2 from about 30 sites, and we use the controls of PMMOV and bovine coronavirus. We were measuring human parainfluenza, but we paused that so that we could run the H5 assay, and we wanted to quickly pivot to that due to the national outbreak and be responsive to a need for providing more data on that outbreak to help respond to it across the United States. Next slide. So as many of you know from attending the stakeholder calls and our communications, we are going to be able to continue WastewaterSCAN for another year, starting July 1st. So on the left side of this plot of this graph or graphic shows the map of all the WastewaterSCAN sites as of today. I already described the number and serving population of all those sites. And then on the right are the plants that are part of the next phase. And so the difference is that we're going to be working with only 148 plants moving forward. The sites that are leaving the program are primarily from California, where 30 sites will be leaving the program, and then a few sites from other states. We will still be serving close to the same number of percentage of the US population, about 12%, and we will still be working with 40 states plus Washington, D.C. So we're very excited to continue working with many of you moving forward. Next slide. So one of the tenets of our program is that we have open and non-proprietary protocols and open access information about our methods. Some of the protocols are on protocols.io, the pre-analytical and analytical methods. We also publish all of our work so that we are very transparent about the science that supports everything we do. And so you can access preprints and peer-reviewed publications on all of the targets describing exactly what we do and how we analyze the data by clicking or taking pictures of these QR codes. Next slide. In addition, I wanted to announce 2 new papers that have come out in the last month. The first is the paper published in ES&T Letters, which is peer-reviewed about detection of H5 marker in wastewater in select locations across the country from Hawaii to North Carolina to Texas. And it's open access, so we invite you all to look at that paper, and maybe somebody can put it in the chat. And another paper that was just published using WastewaterSCAN data is a paper on public health policy impact evaluation, a potential use case for longitudinal monitoring of viruses in wastewater at small geographic scales in PLOS water. We also have on our website a description of all of our published body of work, and I'll show you on the next slide what that looks like. Basically, we have dozens of papers that are the foundational science that this program is built on. And instead of directing people who may not be scientists to actually read the papers, we have short descriptions of the papers with key results and key findings to help you navigate that. So if you go to the website, you can see that little link about publications, and there is a really nice narrative there describing all the publications and the key findings to help you navigate that, so you don't have to feel like you need to read 40 papers to understand what we're doing here. Next slide. Okay, so I'm going to pass it to Marlene, who's now going to talk about our work on H5 detection.
Okay, hi everyone. Good to see everybody. I'm going to talk about sort of the rollout now of the H5 assay to all the WastewaterSCAN sites. If you were here for the last meeting, you can also, happy to have you go back and look at that recording. We talked a little bit about the research that we did and the paper that was published recently demonstrating the foundation for this testing and the work that we've done through the process that we do for all of the targets that we add to WastewaterSCAN to make sure that our process is working really well and we're getting good information from wastewater that we can then pilot, scale, and roll out. So this is just a reminder of the assay that we're using. So working with Verily, we designed and validated an H5 specific assay. So this is for H5 influenza. It's not quite as specific as H5N1. It would detect other H5s that are circulating, but H5N1 is the main outbreak of concern in the United States right now. The assay that we use for influenza A overall is this assay. It targets the M gene. It detects all circulating influenza A, and that would include H5 influenza. And then this new H5 assay targets the HA region. It's H5 specific, and it's about it matches perfectly to about 90% of circulating H5 in the past year. So it should be pretty sensitive to pick up H5 that is circulating, including the clade that is implicated in the current HPAI outbreak in cattle. And all of the details of this assay and its validation and its application and a first few sites are included in that paper, which maybe Allie or Amanda can put the link again in the chat. So as you know, because you've been getting a lot of emails from us about it, we were able to roll this out to prospective monitoring, including the H5 marker in all of the testing for WastewaterSCAN, starting on May 15th. And then about 2 weeks later, we made that data available on the publicly accessible dashboard. And so we are now testing every sample that we receive for WastewaterSCAN for H5. This is the heat map that shows results and some of the states where we have had positive detections. And this map on the left here shows where those have been. So the dark shaded states are states where we have had at least one detection of H5 in wastewater. And the red sites are colored in red are the sites where we have had at least one detection of H5 in wastewater since we started doing prospective monitoring on May 15th. So these are the 6 states where we have detected H5: California, Idaho, Iowa, Michigan, Minnesota, and Texas. As of now, 5 of the 6 have also had confirmed herds infected with H5N1, California being the exception. This is a very recent detection here in the past couple of days. I think something that is notable is that we've seen in a few cases, particularly in Minnesota and Iowa, that these detections have preceded the announcement of herds that are infected by 1 to 2 weeks. And that was also what we saw in Texas when we did some of our original research. The first samples that tested positive were about a week before there was illness in cattle identified, although we were doing that work retrospectively. So we are considering right now that this is very interesting and hopefully useful high-level context information, because of course we don't have complete coverage of every place around the country. And we know that the sort of what is represented in wastewater flowing into utilities and being sampled is very complex. So it's something that we're continuing to work on, and hopefully we would really like to talk more with those of you who are in states and with utilities that have had positive H5 tests, to learn even more about your sewer systems and the different inputs. I will talk more about different inputs in just a second, but I want to point out that we did put together this FAQ sheet for translational support. So we can share the link to this. It will be also in these slides again when you receive it. And so you can see that we just have answers to a bunch of questions that you might be wondering about or that you might be asked. This is something that we can continue to update as we learn more, and as you let us know what questions you have or what questions you might be hearing from others. We would love for you to pass those on to us so that we can update this living document and make sure we're answering the questions. I see in the chat from Michelle, the herds were tested independently, not because of wastewater surveillance, to the best of our knowledge, but my knowledge is also not complete about the workings of state governments. Okay, the last thing I'll kind of say right now, and then we're happy to answer some more questions after the presentation, is about the possible sources of H5 influenza into wastewater. So there are, it's a complex outbreak. It's a challenge and an opportunity we have here. It's a challenge because we know that it's not just human contributions that are likely to end up in sewer systems and produce detections of H5 influenza, but it's also an opportunity because it means we have been able to see really consistently useful information so far about H5 from municipal wastewater. So we're continuing to learn more about what those inputs might be. A really kind of leading hypothesis for a lot of the inputs we see has to do with allowed and permitted discharges into sanitary sewer systems of animal byproducts. Now, this is primarily, we're thinking about milk here, we're thinking about dairy operations, cheese making that produces large amounts of whey that is then permitted to discharge into the sewer system very well can contain high concentrations of the virus. And there are multiple cases where we can see that those industries are there and are contributing. And so we have confirmed in some of our research by looking at upstream samples that there is H5 influenza that has come directly from dairy operations. So Shannon, that's what I mean when we say kind of confirmed that we've looked there and we've seen that that is a contributor into the system. It is also possible that there are other industrial discharges that could produce or could bring animal waste carrying H5 influenza. For example, if there are surface water ponds that are used by industry for whatever their industry is, whatever their process is, and they are permitted to discharge some of that kind of surface water or storm water. So there are other possible non-dairy related industry contributions, but dairy is a big suspect here. There's also possible that there could be infected wild animal accidental inputs into the process. This is probably most likely only if your actual headworks are uncovered or, somewhere before the sampling point in the wastewater system is uncovered and has animals hanging out around the area, right? So this is unconfirmed. We think it's less likely than some of these industry inputs. It's also very possible, especially if there's combined sewer systems, that there could be stormwater runoff, other runoff that is entering wastewater systems through there. And so that is possible. We have relatively fewer systems within WastewaterSCAN that are combined sewer systems. We also know that there are some mostly sanitary sewer systems that have some element of the system that's combined, or there are some more complicated situations. So again, if that's you, we'd love to talk to you more about, your unique systems so that we can understand where these possible other animal inputs could come into the picture. And then finally, it is of course possible that there are infected human inputs into the sanitary sewer system. However, we don't expect that this is likely. We don't have, any direct link that we have observed to any human cases in wastewater systems. And yes, now go, Allie just answered this question. We did. Okay. So I'm going to stop there and pass it over to Amanda to talk about some of our other, oh, wait, I'm sorry. I'm not going to stop there. I am going to quickly show you where you can find this on the website. And so we'll go to the WastewaterSCAN dashboard for a minute. Amanda has put together a really amazing set of slides walking you through where to click on the desktop version and the mobile version of the website to access all of this data. So when we send out the slides after this meeting, take a look at those, and hopefully they'll be a good reference for you should you not be able to follow my quick talking right now. But I'll just quickly show you where to access the H5 data because it is not currently available on this overview page. So when you land on data.wastewaterscan.org and you see this map, if you want to see H5 data, the first thing you'll do is click on the chart logo. So that will take you to this page, and I'm just going to remove that pop-up by clicking anywhere on the screen and go to influenza. So I can choose which pathogen. I'm choosing influenza. And then I'm going to select the H5 subtype. After I've selected that, I can go and select what sites I want to see. And I'm just going to pick on Michigan for right now. And what you'll see is that it will load the heat map view as default. So this will show us the blue squares are non-detect samples, the red squares are detection of H5, and all of the white is time periods where we don't have samples. So you can go through and you can add whichever locations you would like, and you can also look at the data as a line chart. So you can see the exact measurements, although for a rarely detected target, we typically advise people to look at the heat map first, because what we're really looking at here is sort of how frequently and how consistently is H5 detected. And we don't want to over-interpret the exact concentrations, but you can view it in both ways. This little gear icon, as a reminder, gives you advanced settings, so you can add in the raw data points. You can also add in the error bars. So there's a lot that you can do when you click around with this menu. The last thing that I'll show is that if I go to subtype, I can look at one location at a time, and I can actually look at multiple targets at once. And so I can look at influenza A and H5 for this location. And you can see that the results that we've gotten for influenza A and H5 in most cases are very similar. Okay, I am going to stop there, and now I'm going to hand it back over to Amanda, and then we can answer more questions.
Yeah, so we are running short on time, so I'm just going to give a brief overview of SARS-CoV-2, but just a reminder, we will be sending out detailed slides with the national and regional updates. So as a reminder of what the wastewater categories are, we look at trends, levels, and frequency of detection. Next slide. So trends tell us how things are changing, levels tell us whether cases are relatively lower or higher, and frequency of detection is more important for our rarer targets, such as H5 and Mpox. Next. So we have three archetypes of categories for how we calculate the categories. So for SARS-CoV-2, norovirus, rotavirus, parainfluenza, they are commonly detected regardless of season. Then we have seasonal pathogens such as influenza A, B, RSV, human metapneumovirus. And then we have separate ways that we calculate for the rarer pathogens such as Mpox, EVD68, CRS, Hep A, and H5. Next. So I'm just going to give you, like I said, a high level overview for COVID currently, and briefly go over these categories. So currently, the national aggregated population line that we have for all of the wastewater scan sites is in the high wastewater category, and that is due to upward trends and medium concentrations. We've had a 96% detection rate for SARS-CoV-2 in all samples across the program in the last 21 days. Influenza A, B, RSV, human metapneumovirus are all no longer in a seasonal wastewater onset. So we determine onset by whether 50% of our sites are have met that threshold for being an onset. And currently we have pretty low detection rates for all of these pathogens. EVD 68 is in a low wastewater category due to low positive detections. We're only having a 23% detection rate for EVD 68. Neurovirus is in a high category, and that is due to a downward trend, but still high concentrations. The median concentration over the last 21 days is 8.7 million copies per gram, so still very high. Rotavirus is in a medium category. Even though there is no statistically significant trend over the last 21 days, we are seeing medium concentrations and a 97% detection rate. And lastly, for Mpox, C. auris, and Hep A, we're seeing very few positive detections. We haven't had any Mpox detections over the last 21 days, and only 3% detection rate for C. auris over the last 21 days. Next slide. So here's an overview currently for SARS-CoV-2. As I mentioned, it is in the high category due to medium concentrations. And the plot on the right is showing the population-weighted average lines for all of the regions, as well as the national line that we have. So national line is in that gray-black line. You have the Midwest in red, the Northeast in lime green, the South in kind of a turquoisey color, and then the West in blue. So as you can see, we had a big peak around the holidays, end of the year, and then concentrations tapered off, and they are starting to rise again. So that is what we are seeing. The medium concentrations over the last 21 days is 65,000. In May, it was around 44,000 copies per gram. Next slide. And here is a brief overview of sequencing. So as Ali mentioned earlier, we are doing sequencing for about 30 sites in the program. And this plot is showing the aggregated levels for the national program. And over the last couple of weeks, we have started to see more of the KP variants make up a larger portion of what we are seeing. So KP1 currently 13.6%, KP.2, 23%, and KP.3 is 7.2. And as a reminder, sequencing data lags what you normally would see on the site for each individual sample. So this is from 1 to 2 weeks ago. So this data is representative of the week of June 1st. And here is a quick map of where we are at for categories for SARS-CoV-2. 48 of our sites are in the high category, 56 are in medium, and 70 are in low. And currently we have 17 sites that do not have calculations just due to slow samples being sent into the lab. And I believe this is, oh, we have one more slide. So we have a request. I know we mentioned this before, but we would love to highlight the work that all of our wastewater treatment plant partners do. We've been able to highlight some work from Florida, Minnesota, Colorado. And so our request is, if you have photos and you want your story highlighted, please reach out to us. Kyle's email is included on this slide, and we will also be sending out these slides. So if you can't capture his email right now, this will be sent to you later today. And Ali added it to the chat. Thank you. And that is what we have for the slides. We're happy to answer any questions people have now.
So, okay, there are two questions in the chat. I'll take the second one. Michelle, for the next year, we only have the resources to work with about 150 plants. So that's why we have the drop-in sites for the upcoming year. And that's something that we've been discussing on the partner calls and for a while now. So thank you for that question. And then I think we have another question about sequencing. Marlene, would you like to take that question? Or Amanda?
Sure. I mean, I think it's a relatively short answer. I don't think we personally have more information on these variants and their any differences in transmissibility or shedding differences that it is important to continue to understand for our interpretation of the wastewater data, but also a little outside of the kind of our purview. So I don't have a lot of updates to share on that right now.
And Michelle, thanks for your question about how we selected which sites to drop. Actually, there was some self-selection. I'm not sure if you've been on the partner calls before, but we had a survey that was sent out, a very short survey to all of the partners for people to indicate whether they'd like to continue and to what degree they'd like to continue. Most people, I mean, almost everyone said they did want to continue. And we wanted to be able to keep representation across the nation. We wanted to keep high population coverage. We wanted to serve underserved communities. And we also wanted to make sure that the plans that we weren't going to continue working with directly had options for enrolling in state programs or the news programs. So those were all the sorts of things that we considered as a group in helping to figure out which sites that we could continue to work with. And if we do end up having more space, we are going to be reaching out to people. But yeah, that's how we did it.
And just a comment about data continuity. So all of the data is going to stay on WastewaterSCAN.org or data.wastewaterscan.org. So you will be able to access that going into the future. It'll be there.
And I think the question is also like, which sites are being not continuing. And I think just to be clear, everyone knows, and we've contacted personally to discuss the next phase and their participation in the next phase. So we don't have a specific list of the sites, but we have been in very close contact with everyone, and they should know. And if you don't for some reason and you feel like you should, please reach out to us. But somebody at the plant or the local public health department knows, and the CDC also knows as well. And Marlene, do you want to take the H5 question?
Yeah, so I was just typing a response, but for the heat map, the detection is determined by whether or not there has been any detectable H5 in the sample. So whether that is, a certain number of copies per gram, or it is the value of that number of copies per gram normalized by PMMOV, if it is detected, that is expressed on the heat map. Now, if you go to the other part where you can actually plot the concentration of the data, you have the option in that little gear menu to view it either as copies per gram of wastewater solids or copies per gram normalized by the PMMOV measurement. But we don't set a threshold that's different for if it's normalized or not with PMMOV for detection. If it's detected, it's detected, and the PMMOV normalization will just impact then how that is expressed quantitatively alongside other data. Colleen, the detection in California was a relatively low concentration, but well above the limit of detection. I think that's something that's important for us to keep in mind as we think about these different possible sources of H5 into wastewater systems. That We expect maybe if there's a dairy that's handling a lot of milk that has H5 influenza in it, that there would be very high levels of H5 and high levels of influenza observed. But we actually have seen, for the most part, the H5 concentrations that we've detected in wastewater haven't all fit that archetype of being really high concentration samples. Of course, we're detecting influenza in those places. And then we've had those H5 detections, but the concentrations are overall have not necessarily been high across the board.
We've reached our end of call time, and so we are going to stop.